Hello everyone, I am Eugenia Trejo and I'm going to present the work titled Sustainable Synthesis and Characterization of Novel Polymeric Biohydrogel Sargassum-Derived Nanocellulose Composites for Removal Emerging Contaminants. This work was realized in Centro de Física Aplicada y Tecnología Avanzada at UNAM Querétaro. This research explores the green synthesis of a composite material using natural deep eutectic solvent based on polytaconic acid and mixed with nanocellulose from Sargassum species from Quintana Roo, Mexico. The material offers eco-friendliness, cost-effectiveness, and excellent water retention properties. Characterization through various analyses shows its potential as an efficient removal system for emerging water contaminants. This innovation opens new possibilities for composite materials to be applied in water treatment. Respect the methodology, the composite material synthesis consists first in the extraction of nanocellulose from sargassum biomass. This was undergone by physical and chemical processes like alkali and bleaching treatment to obtain it. After that was proceeded to preparing the deep eutectic solvent, choline chloride glycerol, and the natural deep eutectic solvent, choline chloride itaconic acid, by thermostirring method at 60 degrees Celsius. Next, the two nanocellulose samples extracted of flutans and natans species were dispersed in the deep eutectic solvents. The amount of these added was based on the maximum solubility of the solvent. Once the nanocellulose was dispersed and the mixture was homogeneous, the monomer hydroxyethyl methacrylate and the cross-linked bisacrylamide were added. After that, the natural deep eutectic solvent was poured to the mixture. Subsequently, an ammonium persulfate aqueous solution was incorporated to start the free radical polymerization. And finally, the sealed flask was left for 18 hours at 60 degrees Celsius. After the polymerization reaction was completed, the polymer was washed with distilled water and dried at 37 degrees Celsius in a vacuum oven. Then, the emerging contaminant removal capacity was evaluated. The obtained biohydrogels were characterized by Fourier transform infrared spectroscopy to identify their molecular structure and determine the presence of nanocellulose, and the nuclear magnetic resonance characterization was made to elucidate the chemical structure. In the infrared spectroscopy spectra, nanocellulose shows oxygen-hydrogen functional group or hydroxyl group, indicating the hydrophilic nature of cellulose fibers. Additionally, characteristic bands from polytaconic acid are observed in the biohydrogels, showed intense oxygen-hydrogen enriching in the presence of nanocellulose, suggesting a stronger hydrogen bond interactions between nanocellulose and polytaconic acid. Due to the deep eutectic solvents, form hydrogen bonds with natural polymers. Also, it can be appreciated that the polymerization occurred properly because of the intensification of the band characteristic of carbonyl groups. The carbon nuclear magnetic resonance analysis was used to observe the effect of adding nanocellulose of polytaconic acid biohydrogels. The figure displays distinct peaks associated with carbons in the biohydrogel molecule. The peaks at 173 and 171 corresponds to the carbonyl groups of itaconic acid. The peaks between 50 to 100 belong to the nanocellulose structure. These are weak signals due to their low concentration in the biohydrogel. Also, the figure shows an inset with the proposed structure of the biohydrogel, where mainly the backbone formation was in the double bond carbon of polytaconic acid. This confirms that the interaction between nanocellulose and polytaconic acid was by hydrogen bonds. The thermal properties of nanocellulose and polytaconic acid nanocellulose composite materials were evaluated by thermogravimetric analysis. The presence of nanocellulose can be corroborated with this since the decomposition temperature is around 328 degrees Celsius. The signals below 180 degrees Celsius is attributed to the water loss from the samples, and additional signals observed are attributed to the decomposition of additional phases such as hemicellulose and lignin. For the polytechnic acid nanocellulose biohydrogel, the first step corresponds to water evaporation absorbed to the hydrophilic polymers. The second step agrees with carbon-carbon bond cleavage depolymerization, leading to the broad degradation intensity compared to the pure nanocellulose. Eventually, to such broader degradation behavior, the third step of degradation corresponding to decomposition and combustion. The figure also showed that the composites materials increased thermal stability after adding the nanocellulose. In the case of the swelling capacity was evaluated, and it can be observed that the biohydrogels are hydrophilic due to their structure and ability to form hydrogen bonds. The swelling behavior of the samples under different pH and temperature conditions is shown that the polytechnic acid biohydrogels has a lower swelling capacity than counterparts containing nanocellulose. Swelling is influenced by temperature and pH, with the highest swelling at 40 degrees Celsius and increased swelling in alkaline pH conditions. The nanocellulose presence and its fibrous structures in polytechnic acid nanocellulose biohydrogels contribute to improve their swelling capacity. The mechanical properties was determined by tensile tests according to the standard ASTM. The results suggest that the presence of nanocellulose serves as a high hydrogen bonding agent, changing the mechanical behavior of the material. Thus, the strain was decreased for both biohydrogels with nanocellulose are consistent with the increment of the elastic modulus of them respect to the material without nanocellulose. For example, for one of them is almost 5 times superior, due to the materials becoming more rigid and resistant. This is consistent with the thermogravimetric analysis, where the materials with nanocellulose increases the thermal stability. Finally, for the evaluation of emerging contaminant removal, the samples were put in contact with a solution of predetermined concentrations of the contaminants. 3 emerging contaminants were used to study. One of them was a non-ionic surfactant, Triton X100, an antibiotic, ciprofloxacin, and an herbicide, glyphosate. The quantification was made by using UVB's technique at wavelength characteristic of each contaminant. The experiment removal kinetics was determined by measuring the concentration variation of the problem solution at different times, with initial concentrations of 10, 50, and 100 milligrams per liter, varying the pH from 4 to 10 and the temperatures from 5 to 40 degrees Celsius. The mathematical models used to study the removal kinetics were the pseudo-first order and the pseudo-second order, constructed from the obtained experimental removal data, resulting that the pseudo-second order model predicted the experimental load at equilibrium values better. This kinetic result indicates that the emerging contaminant absorption occurs by chemical and physical processes, since commonly occur simultaneously. Besides, the Langmuir and Thornley absorption isothermal models were applied to elucidate the absorption mechanisms. These biohydrogels' maximum absorption capacity and the removal efficiency percentage were determined for each emerging contaminants. Maximum absorption capacity values indicates that the materials with nanocellulose extracted from Flutan's sargassum has the higher removal capacity for ciprofloxacin and glyphosate. For the case of the Triton, the material without nanocellulose expressed the major capacity. The Langmuir parameters suggest that the polymers perform better at higher concentrations than at lower ones. This was supported by the removal efficiency results, which show that it decreases when the concentration is below 100 milligrams per liter. Although having a lower maximum absorption capacity, the material with nanocellulose extracted from Nathan's sargassum showed the best removal efficiency for removing ciprofloxacin and glyphosate. For the case of the material with nanocellulose extracted from fluid and sargassum was the most effective for removing triton at low concentrations. The isothermal model results showed a better fit with the linear isothermal model, which could indicate that the absorption tends to be reversible monolayer process and means it occurs mainly by physisorption processes. By conclusions, the synthesis and characterization of composite material were successfully achieved using a natural deep eutectic solvent based on polyethaconic acid, mixed with nanocellulose sourced from sargassum. The presence of nanocellulose in composite materials enhances swelling capacity, thermal stability, and mechanical resistance. The synthesis biohydrogels proved to be a good alternative for emerging contaminant removal. All of them were efficient in concentrations over 50 mg per gram, and only the material with the nanocellulose from Nathan's sargassum has an acceptable remotion efficiency in lower concentrations. The kinetic model suggests that the physical and chemical absorption processes could co-occur. However, the isothermal models indicate that the removal process is mainly caused by physisorption. Finally, we acknowledge the Cefata National Characterization Laboratory, CONACYT for the Fellowships, Engineering Institute, and Marco Vera for the Nuclear Magnetic Resonance Assays, and the other contributors.